how do you identify a cell in mitosis, in metaphase of mitosis? You look at it. Yeah, the chromosomes in the center, right? Okay, good, excellent. So I'm going to draw what happens during metaphase 1, right, of meiosis. So let's call this metaphase 1. And again, we want to have this understanding that we are looking at processes that increase the genetic diversity of the offspring. Okay? So lining up in the center, except that now the homologous pairs, they line up in a special way that you don't see in mitosis 1. Okay? So again, I will use different color. Now I'm going to just draw blue and red, but understand that there has been crossing over. Okay? Otherwise I will spend too much time. So you have, I'm drawing 2 pairs of chromosomes just so that you know that it's happening to all the other homologous pairs. Yeah. All right, make sure I use the same or the right color. Okay, so you have the lining up of the chromosomes along the center, the small nuclear membrane. But they line up in such a way that the homologous pairs remain paired together. You don't see this in mitosis, right? So there's still pairing. And they line up in such a way, and remember, in mitosis, there's a string that connects something to something. Do you remember that? Yeah. Yes, the centrioles have strings, protein strings called mitotic spindles, right? That connect to the centrioles of each chromosome. Now again, the difference here is it's connected in a special way. So I'm going to draw the centrioles and then I'm going to draw out the spindles connecting. Yeah, so let me do this. So the centrioles are here, opposite ends. So remember that. And then the spindles, like that, and like that. Like that, and like that. So what is the difference now? See if you can pick out the subtle difference. Okay, look at mitosis. Each centromere, remember what a centromere is? Each centromere, how are the spindles connected? Or I should say, how many spindles connect each centromere? 2. And where do these spindles go? Yes, the opposite poles, right? Look at the way I've drawn the spindles. Each centromere, how many spindles are connected? Only 1. And it goes to one end, not the other. Okay? So that is the difference. Okay? Now, why is this difference important? What is the function of the mitotic, the spindles? When the centrioles pull, what happens? It will move the chromosomes, right? So in mitosis, when it pulls, because there are 2, so what happens to the sister chromatids? They separate. But if you look here, if the spindles pull, do the chromatids separate? No, the entire chromosome is pulled to one end. Can you see the consequence of this difference? In other words, when the spindles start to pull, the homologous chromosomes are separated. So sad. Right? Okay, maybe I should draw in the differences just so that you appreciate later on. So let me do this. Right? You can express your irritation at what I always do. Okay, so this is in metaphase 1, right? So the homologous chromosomes remain paired, right? Whereas you don't see this in mitosis. right? Remain.